Chapter 5 Intratumoral Immunotherapy. The cure is within you.
The cure for cancer is within you. There is no better location for the immune system to learn to attack a cancer than within the tumor itself. These new immunotherapy drugs can have powerful effects, but they need to be in the right location. There have been numerous animal studies and some limited human work with injection of some of the FDA-approved immune checkpoint inhibitors, like anti-CTLA4 and anti-PD-1, that certainly are very promising. However, the research world is beginning to move past these into some new areas that look even more exciting. In late January of 2018, a study was published by researchers at Stanford looking at the intratumoral injection directly into the cancer of the immunotherapy agent OX40 agonist antibody combined with the toll-like receptor agonist. The results were outstanding, with cures in all the mice. This led to some very huge news coverage and has set into motion exciting work in humans. We personally are already studying this and similar variations. Systemic treatments with immune checkpoint inhibitors like Opdivo, Keytruda, and Yervoy were just the opening act. Now, intratumoral injections with these other agents is primed to be the closest. There are far too many of these agents to discuss, so I'm going to keep this discussion to the ones that I feel are the most important and that have almost immediate clinical relevance. OX40 agonist. OX40 receptor, also called CD134, is part of the tumor necrosis factor receptor superfamily. This is a co-stimulatory molecule and is expressed on activated T and antigen-presenting cells. This receptor is the opposite of many of the other immunotherapy agents we have described that are widespread in current treatment today, such as anti-PD-1 and anti-CTLA4. As described, those agent block receptors that are the brakes of the immune system. OX40 would be more equivalent to the gas pedal. OX40 actually has a dual function. It can remove the brakes, regulatory cells, and also increase T-cell activation, stepping on the gas. Unlike the therapies for the traditional immune checkpoint inhibitors used today, with OX40, we are not blocking the receptor. We are activating it. As commonly found with most of these immune receptors, you'll find these in many locations, and they can function differently based on where they are located. In this case, the OX40 agonist stimulating antibody works in two ways. It stimulates the immune activation, but also binds to regulatory inhibiting cells and results in their destruction. The simple way to describe it is that OX40 agonist removes the brakes and steps on the gas at the same time. As mentioned, the Stanford study created significant media attention. This study points out several key points, one being that for good success, combinations are going to be needed. In this case, it was a toll-like receptor TLR agonist with an OX40 agonist. Another key point is that intratumoral injection into the tumor is also essential, as some of these medications cannot be delivered to the entire body without potentially catastrophic consequences. This has one other advantage. The reduced doses will also reduce the cost. This is very important as we are already seeing that with current treatment methods, the healthcare system could not support the cost. Ultimately, patients are missing out on their chance at these life-saving medications. We are currently treating human patients of multiple cancer types with OX40 CpG agonists injected into the tumor, like the Stanford mouse study. The results have been extremely impressive. We have found that for humans, to get even better results, we have had to add the CTLA4 inhibitor Yervoy. We plan to present and publish our initial results this year. Toll-like receptor agonist. Toll-like receptors, TLR, are proteins that could recognize various nonspecific pathogenic and dangerous substances. As mentioned, immune responses in cells, such as T cells, are very focused, recognizing very specific aspects of the cells and pathogens that they attack. That type of response must be stimulated and developed. TLR is part of the initial immune response known as the innate immune system. When a new pathogen, or in our case, cancer is recognized, the immune system will not have a specific target and needs to recognize general aspects that may be associated with things believed to be dangerous. Then, with the right stimulus, this can convert to a more effective and specific immune response. The best way to understand this is going back to our car analogy. If the other immune agents we discussed are removing the brakes and stepping on the gas, TLR are turning the key and cranking the car. As you can imagine, if the car is not running, removing the brakes and stepping on the gas will have no effect. This is what can lead to failure of the traditional immunotherapy agents. They forget to crank the car. Also, in immunotherapy, it is not only the right combination of agents, but the correct sequence as well. That is vital for success. There are some studies showing that immunotherapy in the wrong order may negatively affect your future potential for a successful immune response. First, you need to stimulate the immune system to be attracted to the tumor. A good example goes back to the Stanford cancer vaccine study, in which the researchers used the TLR to get the immune response started, then ramped it up and further activated it with the OX40 agonist. As you can see, one without the other and your car is not likely to go anywhere. It was this combination that proved to be highly effective in the study. For an effective immune response, the immune system not only needs to recognize cancer, it needs to know that it is dangerous and should be eliminated. One key aspect to TLR agonists is that they will attract the immune response to the area they are located. This means that you cannot use the drug systemically throughout the body, such as oral or IV administration. These need to be used directly at a tumor site. As I have mentioned, most drug companies prefer systemic administration as it is easier to give. Oncologists are not trained to inject tumors, and they are the ones that control most cancer treatments, and certainly drugs injected into tumors will generate less revenue than ones given systemically. There is an FDA-approved topical TLR agonist called imiquimod, trade name Aldora. This is used in certain skin cancers and is being evaluated for topical use in breast cancer as well. In the Stanford study, they looked at the direct injection of a TLR9 agonist called CpG and a TLR7/8 agonist called redefining. CpG are short single strands of DNA that are found in high amounts in certain bacteria. The immune system has evolved to recognize these as being dangerous, which helps activate an immune response. This is considered the main active component in Coley's toxins, which were killed bacteria that Dr. Coley injected into cancers to help stimulate an immune response against cancer over 100 years ago. Think of CpG being the most refined version. This is an important aspect of generating an immune response. However, in most cases, it was not enough to do it alone. There was something missing to take the immune response to the next level. Ultimately, there may prove to be several immune agents capable of advancing the initial immune response of TLR agonists to a durable cancer cure. OX40 agonist looks like it could be one of those missing pieces, at least in some cancers. In the war against cancer, one important and forgotten aspect is getting your troops to the right location. If there are no troops, then activating them will do no good. This is one of the main reasons traditional immunotherapy may not work. The tumor environment does not have enough immune cells present. When immune cells are present, we call that an inflamed or hot tumor environment, which has a better prognosis. When immune cells are not present, we call that an immune desert or cold tumor environment, which has a poor prognosis. There needs to be signals to attract the immune cells to the tumor. However, this is even a little more complicated than you might think. The immune cells do not just swim to a tumor. The cells lining blood vessels must summon them. Since tumors control local vessel growth, you may guess that for tumors to survive and avoid the immune response, they build vessels that are less likely to attract the immune cells. They actually have vessels that may bring in regulatory cells to protect them and destroy the effector, attackers, T cells that want to kill them. Essentially, the tumor controls the roads in its territory, so it will keep out the enemy. This is another barrier to the immune system. A great review article published by Johansen Percival et al. In Trends in Immunology, Immunomodulation of Tumor Vessels, It Takes Two to Tango, is an excellent overview of the current understanding of this process. One reason I mention this here is that it has been shown that CpG can help initiate changes in the vessels, which can then attract the needed immune cells. Think of the immune cells being on a train that can only get off where there is a stop. The cancer instructs blood vessels to form without stops for the attacking immune cells so they can get off. CpG helps make a stop so the immune cells now can get off where the tumor is located. As you can imagine, you can do everything possible to stimulate your immune system with the typical immunotherapy drugs, but if the immune cells never get to the tumor, the therapy cannot work. This further supports the critical importance of intratumoral injection of CpG. The other agent they mentioned in the article that can help is a low-dose anti-VEGF drug, bevacizumab, Avastin, which we will talk more about later in this book. STING agonist. Stimulator of interferon genes, STING is a protein that recognizes foreign pieces of nucleic acids, DNA, RNA, often found in cells that are infected with pathogens such as viruses. The recognition of these foreign nucleic acids triggers an innate immune response with secretion of immune-stimulating cytokines such as interferon. This has a lot of similarities to the previously mentioned TLR agonist and overlaps with Coley's toxins as well. These STING agonists set up an initial immune response which can ultimately prime the T cells to be ready. As mentioned, it is the T cell response that leads to a more specific and durable anti-cancer immune response. The T cells are the main target of the currently approved immunotherapies such as anti-PD-1 and anti-CTLA-4. Again, if the T cells are not recruited and stimulated in the local cancer microenvironment, then these popular approved immunotherapies are unlikely to be effective. It is for this reason that agents stimulating an initial innate immune response, such as TLR and STING agonists, are gaining a lot of attention. In cases where the initial immune response is absent, to use current immune checkpoint inhibitors is essentially putting the cart before the horse. In addition, as I mentioned with the TLR agonist, CpG, it seems that STING agonists can also cause changes to the vascular structures which enhance tracking of the immune cells back into the tumor environment. It also creates those new train stops for the T attackers. Glenn Barber, PhD, from the University of Miami, discovered STING in 2008, where he showed that mice lacking STING were very prone to viral infections. Later, Russell Vance, PhD from UC Berkeley, published in Nature 2011 that cyclic diguanylate monophosphate found in bacteria activates STING and stimulates an initial innate immune response. Basically, as later discovered by Jujian Chen of the University of Texas, Southwestern, there is an enzyme that links these nucleic acid pieces into a circle that stimulates STING. Normally, our own DNA would be located in the nucleus and would not trigger this response. This has now led to the development of synthetic cyclic dinucleotide, CDN, or other small molecules to activate STING. There have also been animal studies showing that the combination of TLR and STING agonists seem to work together, further enhancing the anti-cancer immune response. Certainly, the STING agonist may have a bright future in cancer immunotherapy, but currently it is not as developed as a TLR agonist. Even though the drug companies hope to develop a STING agonist that can be given systemically, most of the initial work shows the promise is with the intratumoral injection. I suspect the real benefits will come with combining both the TLR and STING agonist injected directly into the tumor, still with other immune agents such as OX40 agonist. One thing to note with STING agonists is that there are human and mouse versions of STING. Researchers were studying a STING agonist in mice called DMXAA. It looked extremely successful, but did poorly in human trials. This allowed them to realize that some substances, such as DMXAA, may only bind to the mouse STING. The synthetic cyclic nucleotides, CDN, that were first studied activated STING in both humans and mice. However, researchers have been trying to find other small molecules that may be given in a different method than just injecting into the tumor. I understand their reasoning, but intratumoral injection will continue to be the safest and most effective route of administration of drugs such as STING and TLR agonist. In addition, researchers from Tufts University, Larkin et al., J. Immunol 2017, showed that high doses of STING agonists can have the opposite than desired effect and cause T-cell death. So, as you can imagine, it will be hard to create a pill or IV therapy that would not give someone too much of the drug, since you must flood the body just to get enough in the locations of the tumor. This further supports that case that intratumoral administration should be superior. As I was writing this book, a publication by Zhang et al., Identification of alpha-mangostin as an agonist of human STING, was released in October 2018. This is an exciting development. They also describe that mangostin may help convert tumor-protecting macrophages into tumor-attacking ones, another key area of immune resistance. It is interesting as mangostin comes from the fruit of the mangosteen tree, not to be confused with mangoes. It is nice to know that this potentially very important cancer medication can be derived from a natural fruit. It is unclear if just eating the fruit would have much benefit or not. If it does, I suspect it would be for a different reason than the STING agonist. A 2014 thesis by Dr. Fabiola Gutierrez Orozco, Ohio State University, indicated that mangostin extracts ingested orally may have a negative effect on the microbiome, increasing inflammation and reducing bacteria diversity. This makes sense, as it is to be expected that it would create a direct inflammatory response, attracting the immune cells. This is something you want in your tumor, but not necessarily in other areas of your body, like the colon, further supporting the direct injection approach. Intratumoral injections of vaccines, viruses, and vaccine adjuvants. There are numerous techniques to stimulate an immune response into a tumor. It has been noted that in certain cases, vaccines intended to prevent other illness may inadvertently stimulate an immune response within cancer. Myers et al. Published in March 2005 an article, Oncolytic Activities of Approved Mumps and Measles Vaccines for Therapy of Ovarian Cancer. They state, in the study we demonstrated that two commercially available paramyx survival vaccines, Morton measles and Jerry Lynn mumps, have promising anti-tumor activities against human ovarian xenografts established in the peritoneal cavities of immunodeficient mice. Galanis et al., also from Mayo, published January 2015 in Cancerous that the use of the Edmonton vaccine strain of the measles virus showed effectiveness in the treatment of ovarian cancer in patients who have previously received extensive chemotherapy, extending survival to 26.5 months in a group of 16 patients. Imsaul et al., also from Mayo, reported anti-tumor activity with the same Edmundston measles vaccine. I might add that this is the old version of the measles vaccine. Curiously, about the time of some of these publications, these vaccines have become hard to find, and it seems their sales in the US have been blocked, as now everything has shifted to the use of MMR in place of the older vaccine. I won't speculate the coincidence of the timing of this. As you see from the study, the results are not perfect, but I am sure the success can be increased with the combined use of other immunotherapy agents. In July of 2018, Mackenzie and Nichols published in JAMA Dermatology a study in which a 97-year-old woman with squamous cell carcinoma of the skin was treated successfully with a series of injections of Gardasil, the HPV vaccine. They initially injected the patient in the arm to initiate an immune reaction and then follow up with the injection into the tumor. They noted that non-treated lesions resolved as well. There are numerous viruses or viral-based gene therapies that can be injected into tumor and can stimulate an immune response. Like most treatments, the use of these alone is probably not enough to generate a cure in most patients, but this can probably be significantly improved when used with the right combinations of immunotherapy agents. It is also reasonable to mention that, just like in vaccines, for the combination of immunotherapy agents to be effective, they often need a carrier agent known as a vaccine adjuvant. This could be a key aspect to generating an immune response. In our work, we use Montanide ISA51 or saponin-based vaccine adjuvants, sometimes with a gel delivery. The idea is not only to enhance antigen delivery to the immune cells, but also to create a slow-release formulation, keeping the medicines in the tumor environment. Montanide ISA51 is used in several vaccines as an adjuvant because it is a water and oil-based mixture that enhances antigen delivery. It seems that the oil can solubilize some of the medications and create a slow-release delivery system. Another promising intratumoral therapy involves the use of gene therapy that causes the increased production of interleukin-12 within the tumor. IL-12 can activate both natural killer cells, NK, and attacking cytotoxic T cells. Studies are being done to inject the gene in plasmid within the tumor. In addition, the gene can be placed within a virus and injected into the tumor. In this case, both the virus itself and the increase in production of IL-12 can stimulate an immune response against the cancer. In addition, there are a few other immunotherapy agents that are being studied, which all have excellent potential alone or better yet in combination with other previously described agents. These are the ones I am particularly interested in: CD27, CD40, and CD13741BB agonists. We are currently working with these and looking at the combinations with other immunotherapy agents. Like I said, we have most of the weapons needed. We just need to learn more about the right combinations. As you can see from this chapter, there are numerous therapies that can be injected directly into the tumor to stimulate an immune response. These combined with some of the other traditional immunotherapies, such as immune checkpoint inhibitors, may hold the key to highly effective cancer cures. In addition, because they are directly injected, they can reduce the number of needed treatments, side effects, and overall cost of therapy, which is a win all the way around.